Hi, good afternoon, good morning, good evening, wherever you're joining us from. I'm Birgit Meldal. I'm the Senior Curator for the Complex Portal. Thank you, Ajay, for the introduction. And I have also with me Elliot Ragenau from our team. Yeah, so Elliot will moderate the chat, or rather the Q&A. He will try and answer your questions where possible while I'm presenting, but I will take some breaks in between if there are more general questions to be answered to everyone. If you participants want to use the chat, be free to do so, but we prefer that questions to us go into the Q&A so it's easier to handle. So with that, I will start. Hopefully we have a few more joining us in a minute or two. There we go. All right, so we are presenting the Complex Portal today. So I'm starting with explaining why we actually use the Complex Portal. So this is a database bespoke for macromolecular complexes, and you might think, oh, you know, there's complex data in many places. Can we not find it there? And you're right, there's complex data in, for example, the gene ontology. It provides us with descriptions and definitions of complexes, and it provides us with annotations to gene products that are related to these complexes. So, for example, in the proteasome complex, there are over 400,000 annotations in the gene ontology to proteasome complex, and this range over a whole range of species. Even if you go and filter by human, you still have over 800 annotations. And these annotations are all to proteins and gene products, but that's more annotations than there are proteins in the complex, in the proteasome. So how do we get more details about the members of the proteasome? Clearly not through the gene ontology annotations, but we can take this term from the gene ontology and search UniProt. And then in UniProt, we can again filter by human and also just the reviewed proteins in the SwissProt data part of the database. We get 63 proteins that are annotated to the proteasome term. But that's still more than the proteins, unique proteins are found in the actual proteasome complex. And you can see towards the bottom of this screenshot, there are some associated proteins like RAG23 homolog B or the insulin degrading enzyme. So that's still not close enough to characterizing the whole proteasome. 4 complexes that have been identified by or characterized with crystallography or electron microscopy, you can find these in the PDB consortium. So there are now full structures of the proteasome. They are proposed to be made out of 32 distinct proteins and make 60 macromolecular structures. And the PDB gives you a link back to UniProt and all the identified proteins. But that's very specific for complexes that have been identified, complexes that have been crystallized. So a lot of complexes haven't been crystallized. Where do you go from there? For human complexes, you can also get information from Reactome, but that's human-centric and again, doesn't provide all the information you might need, although it does have a list of members. You can go to Intact or an IMIC consortium partner, and you can look for molecular interactions, but even filtering for proteasome complex and human, you get over 9,000 molecular interactions. So that's again more than what you need to characterize the proteasome. So the Complex Portal brings it all together. And we provide you, we take the definition, the list of participants, we link to structures and reactions and molecular interactions, and everything we curate is based on literature, on experimental evidence in most cases, and published data. What we provide you, therefore, are stable, unique, and versioned identifiers. and a whole lot of additional annotations in one place. So you don't have to go to all of these other sources to try and identify the complex. So what we're doing is we provide an open access, high quality, manually curated knowledge base of annotations of macromolecular complexes. Okay, and how do we define a complex? So our definition is that it's a stable set of two or more interacting molecules. Typically, they contain at least one protein molecule and have been demonstrated to form a biological functional unit. And this is the important thing. Everything we curate is the functional unit in the cell. Macromolecular complexes don't have to contain proteins. They could be made up of, for example, just RNAs. At the moment, all the entries we have got at least one protein component. But these complexes can also contain non-protein components, like small molecules that often act as cofactors, such as metal ions or nucleotides, and nucleic acids. So any type of RNA or DNA that's absolutely required for the complex to form. So here are some examples. The GARB complex in C. elegans is just made up out of 4 different proteins, whilst the telosome in yeast contains the telomeric DNA. The other 3 proteins do not form a complex without the DNA they bind to. And the Cry1per2 complex in mouse contains the zinc ion as a cofactor. We don't curate simple enzyme substrate associations or one-to-one ligand receptor complexes or assembly intermediates that don't have a unique function or other similar transient interactions. And to put that into context, for example, the mitochondrial respiratory chain complex 1 is made up out of over 50 components, but during the assembly process, there are lots of intermediate steps that are being built with certain proteins that just come in as stabilizers while other components are added and then they disappear again. And so the complex portal only contains the final functional complex 1 with the 50-something components. On the other hand, the complement cascade for complement 3b contains a range of different C3b complexes that all have a function along that cascade of attacking pathogens in the host organism. And therefore, there are a number of different C3b complexes in the complex portal. Finally, we don't curate simple associations recovered from pull-down or co-immunoprecipitation experiments because, again, we don't have the functional information at that point. We have to wait for the functional experiments to be conducted as well, and then we can curate the complexes. There's an exception. These are mainly receptor ligand complexes where the receptors don't form complexes on their own in the inactive state. They're just sitting in the membrane as monomers. These are typically membrane-associated molecules such as tyrosine kinases. And here we curate the ligand receptor complex as one obligate complex. So in this example, and the screenshots are taken from Reactor because they're beautiful and they have a free reuse license. I'll encourage you to use these pictures yourself, these icons. We curated the platelet-derived growth factors, which are obligate dimers as ligands in the extracellular membrane. And once they bind to the receptor molecules, the receptor molecules come together and form this obligate tetramer that you can see on the right here. And we don't curate a receptor dimer out of the 2 receptor chains because they don't exist in vivo. Okay, which organisms do we focus on? At the moment, the focus is on model organisms. So no more. This is on model organisms, and we focus on human, mouse, yeast, and E. coli at the moment. We have now, in inverted commas, completed the yeast and E. coli complexome. That means that we think we've included all the known, well-characterized complexes for these two species. This is obviously an ongoing process. New complexes are being characterized, and we're working with the specialist community, for example, SGD and EcoSci, and adding complexes as they're being newly characterized. The first yeast complexome was published nearly 3 years ago, and since then we have added or updated over 20 complexes already. You can see that there are a number of species for which only a small number of complexes exist. And this is because sometimes complexes have been characterized in a different organism, where for that particular molecular function, this organism is used as a model. So, for example, there are a lot of cardiac receptors and transmembrane complexes that have been characterized in bovine tissue. So we've curated the bovine complexes first, and then we've inferred them to the human. And I'll show you a little bit more about that in a moment. We also, this year at the start of the pandemic, curated the SARS-CoV, SARS-CoV-2, and MERS-CoV complexes, and over 100 human target complexes of these viruses, and that's an ongoing project. In general, we're focusing on human mainly at the moment, but we also have a curator who focuses on C. elegans, and as I said, other things are coming in as they're being requested. We add information on the evidence on each complex using the evidence and conclusion ontology. If experiments are available in Intact, IMEX Partners, PDB, or EMDB, then we link directly to these experiments and we add the code for physical interaction evidence used in manual assertion. And at the top of the details page, which we will see shortly, there is a full reagent flask that indicates that there's evidence for the whole complex, protein molecular interaction evidence. But in many cases, the interaction evidence isn't available for everything, for the whole complex. And so we're using this branch of the eco-ontology called biological systems reconstruction evidence. And we have 3 different situations here. We have cases where, as I just explained, we have experimental evidence from a species that is not our focus. So we curate, for example, in bovine, and then we infer to human and annotate this as inferred by orthology. In other cases, we have complexes that are very similar to each other in the same species, so we can infer them based on paralogy evidence. So these are cases where, for example, a complex made out of protein A and protein B has got experimental evidence. And we know from function experiments that a protein C is very similar to protein B. And then we infer that the complex between protein A and C also exists. And these complexes are identified with a half-full reagent flask on the website. There are cases where the experimental evidence is made up out of mixed species. So experimentalists in the wet lab have made constructs from human and mouse, for example. And then we look at the sequence similarity and other evidence to see whether these proteins in human and mouse are near identical. and take other functional evidence into account to say, yes, although the only experimental evidence we can find is this crystal made out of human and mouse, but we believe that this complex does exist in a proper human and a proper mouse, and then we use that as evidence and give it the full flask evidence code, evidence identification. And these entries are again linked to intact or PDB or EMBL-EBI. And then we have a group of complexes for which the experimental evidence is only partial. So it's partial molecular interaction evidence, there's partial functional evidence, there might be pharmacological evidence that this complex exists, but generally speaking, based on all the circumstantial evidences, the community working in this field believe these complexes exist. And so we basically infer them by curator based on inference from background scientific knowledge. And they're identified with an empty experimental flask. I mentioned we've updated complexes, and for that we have versioning systems. The website and the download files always contain the latest version, but if you go to the XML file, you can see the version number. And the older version are all accessible in the archive on the FTP. Talking about files, we provide you the data not only through the website, but also through 4 different file formats. The XML file comes in 2 different versions. These are based on the community standard, the Proteome Standards Initiative for Molecular Interactions. We provide both standards, but the XML 3.0 is more expressive and contains more fields that are specific to complexes. The list of participants is available in both. If that's all you're after, you can use both of these files. We provide you with a simple tab delimited format that can be easily human read, that can be imported into Excel, and the complex tab is 1 file per species. And then we also provide you with a JSON through our web service. And this JSON is also used for the complex viewer, which we'll see shortly. One quick word about the curation pipeline. It is very similar to Intact, which is why the Intact logo is on this slide. But in addition to the publications, as I mentioned, we bring in information from a lot of partner databases, IMEX partners, Go, SGD, Reactor, Uniprot, PDB, and so on. We bring all this information together. We create an entry in the database. We use controlled vocabularies and curation manuals to make sure everything is standardized and logical. And these terms and manuals are living documents that evolve as technologies evolve, as more data comes in. Once the curator is happy with the entry, they'll send it for checking, and the second curator checks the entries and makes sure there are no mistakes, or it can create a discussion about how best to represent this complex, because sometimes complexes are rather complex. Once the 2 curators have agreed that the entry is complete, it gets accepted, and with our bi-monthly releases, the complex will be available on the website, the FTP site, through the web servers, and a number of other databases that take our data and provide it. The whole curation strategy is based on Intact, and the backend, the file system, the release process is all synchronized with Intact, so the data comes out at the same time. Okay, at this point, I'll take a break. I don't know if there are any questions anyone wants to ask at this point before I go into a demo. Okay, so if there are no more questions, we'll start to look. I welcome you to I need to change this. Right. So I welcome you to have a play around at the same time. Elliot has posted the link into the chat so you can open it in your own browser. This is the front page with a simple search field, which we'll look at in a second, some extra information through the tiles, and acknowledgement of all our contributors on the bottom, as well as our Twitter feed and newsletter subscription. You can get documentation through this tile here. There's a data content section, which describes a little bit more about how we curate, and a syntax query information, which is quite useful. There's a summary of what you can search for, and also specifics about how to search for different types of fields within the complex data structure. So you can keep that in mind if you want to construct some more complex queries later on. For now, we'll look at some simple searches. And you can see some examples here. You can search with all sorts of names or accessions for genes or proteins. If you already have a list of complex ACs from a previous search, you can reuse those, and you can also search with lists of identifiers. Let's start with the probably the most well-known complex, hemoglobin. You might have been the first complex you heard of in school or when you did your degree. And it's a perfect example. So how do we search for hemoglobin? Well, firstly, we can search for the name. And here we already have a problem. Do we use the American or English spelling? Now, I use the English spelling, but I get complex results with the American spelling because we've predicted this, and we use synonyms at the back end in the data to deal with these spelling differences. Now we see that most of the hemoglobins we've curated so far in human, some from mouse and some from rat. But we've got all sorts of specialized hemoglobins at the top of our list, so this was probably not what we were looking for. Now, if you, like me, remember that hemoglobin has got alpha and beta chains, and because I looked at this into a little bit more detail, I know what the gene names are. So that's hemoglobin A, and you could do a Boolean search for hemoglobin B. So now we're using the gene names. And now we find hemoglobin for human, mouse, and rat, the normal adult hemoglobin. Now, a more precise search would have been to use UniProt identifiers. But if you're like me, you don't know the UniProt identifiers from the top of your head. So you could have gone to UniProt, searched for HbA and HbB in human. Now, if we go into the human complex here, this is our target, human hemoglobin A, AB. Each of our entries has got a recommended name, which is generally the name the complex is best known for in the community, but we do give a list of synonyms at the bottom of the page. Each complex has the unique identifiers. Each complex is unique to a species, and we also provide you with the tax ID. Here's the link to the evidence. For hemoglobin, we've got evidence from Intact, and if we click on it, it opens the interaction evidence. And we can see that this is linked to a crystallography experiment, and we could even go through to the PDB entry here. We can download the complex, as I mentioned, in the 3 different formats. The complex tab is not available for individual entries, only for the whole species, and you can get to that by following the link to programmatic access. You can escape this pop-up. You can say, oh, this is interesting, I might need to go back to it, so I can pop the complex in the basket, and it pops it in here. And then you could also remove it if you want to. It's not exportable at the moment. That's still a feature that we're working on. So we go back. Each complex has the list of identified members. And as I said, this is all based on publications. In this case, we know the stoichiometry, we know there are 2 alpha chains, 2 beta chains, and each of the alpha and beta chains are linked to 1 heme molecule, which is a cofactor. So you can see the roles also identified here and links to CABI and UniProt. And in the viewer, you can see the topology. We'll have a look at the viewer in more detail shortly. Every complex is annotated with the functional statement that's written by the curator, very much like UniProt. Every complex is annotated to gene ontology terms as precise as the knowledge for that complex stands. Sometimes we don't know the molecular function. Almost invariably, we have a biological process annotation, and if you click on View All, you can expand all of these lists. By default, only 5 members of a list are displayed. For humans, we link to complexes in reactome. As you can see, there are 4 different types of hemoglobin in reactome. They differ by oxygenation stage and cellular location. So you can click through the different complexes, and you can see on the bottom of the page, the reactome diagram is visible here and highlighted in pink. And if you click on a different complex, the highlight changes. And if you click on a different pathway, hemoglobin is available in 3 different reactome pathways, it changes the pathway. And if you want to find out more, you can click on the reactome link and open the whole pathway in reactome and go deeper into the details there. Many proteins also have an annotation to properties. This can provide more information on internal topology, on cofactor functions, or in this case, we know the molecular weight for it. There might be Stoich's radii information on there as well. Each complex that's completely characterized in terms of their stoichiometry also has an assembly terms, for example, heterotetramer for the hemoglobin. We then add crosslinks to PDB and also EMDB where applicable. These are manually added by the curators and are not deemed as complete. For example, for hemoglobin, there are over 100 entries in the PDB, and we can't add them all manually. So we give you some examples and you can then explore it further in the PDB. We also import this widget from the Gene Expression Atlas that provides you with the gene expression of the individual genes that the proteins are coded by from the tissue expression data. You can hover over these elements to see the details about the expression. We annotate each complex to a cellular component. This is usually a complex term, but where applicable, we can also add the subcellular location. We annotate to diseases and pathologies using the experimental factor ontology that's also held at EBI. So you have a text definition and then the link to the details in EFO. Where the complex is a drug target, we provide links to Campbell. We can provide links to other resources like Senior, MatrixDB, Protein Ontology. There are quite a few options depending on the entry. And references for further readings. In this case, it's the link to the crystal structure. The literature references might go to functional experimental papers or reviews as well. And then, as I mentioned, there's a list of synonyms available and a systematic name that takes into account stoichiometry. Okay. Right, so these are the details on any given details page. Are there questions specific to the details page? No, there's one question here. What are complexes, as most of biological compounds are quite complex? Yes. So our definition of a complex is anything that has a function in the cell, metabolic function. And so most of the time they have got proteins. Everything in the complex port at the moment contains at least one protein. As I mentioned, there could be RNA complexes if they have a function. We don't include small molecule complexes because there are other databases like EBI that are much more specialized for that. I don't know if that answers the question. So they're the biological functional units to carry out a metabolic function of some sort. Okay, and I mentioned we have a look at the viewer. I will use a different example, and Elliot will post the spelling for sheltering in the chat in a second. This complex has got more features in the viewer, so it's nicer to present. I forgot to mention, do have a play in parallel if you have a second screen and explore what I'm showing, so you can ask them more questions at the end as well. So you can see in the viewer here, there are different wedges within the protein symbols. If I hover over the background, it gives me the complex ID. If I hover over these symbols, it gives me some annotations, and they're more visible if I expand these. I can just click on it to expand it, or use the button and expand all. And then I can hover over the regions, and that tells me what the binding region is. These are all manually annotated molecular binding regions based on experimental data. And you can move these around. I should move the other way. Move all of these elements around. Stretch them out a bit. If you hover over the edge, it gives you the information for the binding region for both proteins that it, or both elements that it binds with. It connects. You can further zoom in by clicking on the bar, anywhere on the bar, by the way, and then you can get down to the amino acid sequence. Click out of the complex, you can move it around, and we can get to the actual binding region and see the actual amino acid sequence of that binding region. And you can see the overlapping regions are identified in separate lines. Again, where there's no information about a specific binding region, as in this case, it's annotated with question mark to question mark and the hatched area across the entire protein length. Then, in addition to molecular interactions, we can also visualize some of the selected UniProt features. So now the focus changes. You can still hover over the colors, and it gives you the name of that UniProt feature. And you can see the same feature is highlighted in the same color, and it gives the range, of that feature. You can highlight the domains in the superfamilies. As you can see, those superfamily domains are often very, very closely overlapping with the molecular binding features. If a protein is filled in gray, it means there's no annotation for that particular feature type available. And then you can also highlight the unique participants. And this is not very nicely visible at the moment because I start to expand the protein bars. You can collapse them all. And then you can clearly see, again, the stoichiometry. So we have 2 TERF1 molecules, 2 TERF2, and then one of all the others, which is the same that's in the table here. You can show all molecules here. The legend refers to the symbols in the viewer, they're matching. And you can export the viewer to SVG. The viewer is freely available, so you can export all of those, not structures, all of those topologies for your own further use. So that's all on the details page. I'm just having a look. I will answer the protein structure question later. The tissue specificity is annotated. That one is imported. So the tissue specificity, which is this widget here, that is imported from gene expression. We don't do any annotation on that. It's a widget they supply. And if there are any problems with that, you should send them a message through this link if you think that their annotation is incomplete. I will let you know very shortly how to get in touch with us to complain about misannotations in our pages. Okay, so if we come back to the home page, what else can we do? We can search for everything with the wildcard asterisk. And you can see that you can filter by species, as we saw earlier, but you can also filter by the biological role. So you can, for example, say which complexes just have a regulator in them. And you can see here in this example that each of these three molecules, three proteins that are part of the complex, have got an extra annotation. So it's an enzyme regulator, and a plain regulator. So we can get down to biological role annotations, and you can filter these here. And for some reason, it now filtered by C. elegans. We can reset that filter. And similarly, you can filter by interactor types. So say you want to see what transfer RNA complexes we have here. And then you can see the transfer RNA. If there are only two proteins in a complex, we expand the viewer by default, even if the binding regions are actually unknown. We just know that they bind to each other. Okay, another question. Is it possible to go from an entry page and click on the protein and see if it's another complex? Good question. That's something that we are thinking about implementing, but it's for a future project. What you can do is you can highlight, you can copy an identifier and search with that again. And here you can see in this case, this was not pre-prepared, that's just by chance. There are actually two complexes in E. coli that have this particular protein. Second question from Hector. Is there a way to make a query to rank proteins by the number of complexes it participates? No, we don't have a ranking system for that at the moment. There is, if you go into the documentation, I can never remember the syntax myself, what you can do is filter by number of participants. And I believe it also does a ranking, but I can't remember. So we have 584 complexes that are trimers. I can't remember if it works like this. No. Now I can't remember if you can do it. I don't think you can search for a range, but you can search for all trimers, all tetramers, etc. So what else have we not looked at? This is the way to get to the organism page. I showed you the screenshot earlier. So from here, you can now either click on the icon and find all yeast complexes, or go directly to the download options for the three files. So here's the way to get to the complex tab as well, and then just download it from the pop-up. Okay, another question. Does a interaction binding is annotated from one publication with strong evidence or confirmed from multiple publications with same interaction information? So the evidence for interactions, the primary evidence, experimental evidence, is held in intact. And we are not linking directly to intact for individual interactions. So I just clicked into an example. So there's no way at the moment to show you where in intact you have interactions based on this binary edge. For complexes with interaction evidence, you can click on here, and that will give you one example. The only other way is to actually then do a de novo search in intact with the proteins that are part of the complex and get all of the information from there. I don't know if that answered the question the way it was intended. So that was the organism page. Programmatic access, exactly what I showed earlier on the screenshot. Training links to our training webinars and other training activities within the molecular interaction team. Is there a way to download everything? Not all of it, but if you go to programmatic access and then go here, these files are available from the FTP site. You can then download the complex tab PSI, so that's the XML 2.5 or XML 3.0 per species. So you got per species files and folders. Okay, next one. Sorry if you went into this, but for a given entry, can we extract the amino acid list making interactions of a given protein? Yes, so essentially what you do, and we can go back into an example, we can use the basket. So when you download this individual complex, either as XML or JSON, you will get the list. If you want a simpler version, you need to download the complex tab, but then you have to download the complex tab for the whole of the species, so the whole human complex tab or the whole yeast complex tab, etc. And yes, the PDB will also give you that, but only for those where structures are available, and the majority of our complexes still don't have available structures. Right. So if we go, yeah, that's everything from the home page. I will flip back to the slides for a moment. There are a few other mixed cases in terms of annotations where we have some evidence, but not evidence for everything. So, for example, sometimes we don't know all of the internal binding topology, and then you have these proteins sitting in a bag within the complex. If the stoichiometry is above 20, we collapse it, because otherwise you wouldn't be able to see anything in the view and it wouldn't make any sense. We can drill down to the individual binding sites for cofactors, for example, and also for amino acids. So in this case, the two proteins interact through a per-sulfide bridge, and we annotate it down to the individual amino acids. The view is available, freely available through GitHub. You're welcome to reuse it. You're welcome to contribute to the code. It's a community project. It's written in Java and uses 3D for the position of the nodes. And it's available also through NPM and browser Xerify, and Bio.js. And it's been embedded, as far as we know, in the complex portal and also in the human mine and yeast mine through a collaboration we had with Intermine for a number of years. If you have any questions later, you can always contact us through the yellow tile on the front page. It says Request Complex. It opens a simple EMBL-EBI contact form that goes straight to the complex portal team. You can request the complex, you can request an update, you can report it back, anything. If you have a GitHub account, you can report back straight to it. Please don't send curation requests through GitHub, only bug reports. And you can follow us on Twitter or subscribe to the newsletter. There are things, there's another question. Okay, question. If the only way to have amino acid list for the interactions by structures or also using Fred. So we don't mind what the experimental evidence is, as long as the experimental evidence is specific for the complex, not a large scale pull down or immunoprecipitation study. And the experimental data is found in IMEX partner databases, PDB, EMDB, and we build on top of that evidence. So we don't really mind what it is, and a lot of complexes don't have a structure, so we have to use other type of experiments. A lot of pull down, a lot of column purifications or enzymatic assays as well. Good question. If any publication is controversial evidence for a well-known complex, how is it addressed? Good question. It's either curator judgment. We can put a comment in the definition to say we are not entirely sure if this is the final version of the complex. We have a number of complexes that are annotated with the curator inferred eco code based, because the full list of components is not entirely known yet. So we put in what we think is part of the complex, but we might add comments about proteins X, Y, Z, and we add the UniProt cross-references to it, may also be part of it. So we're kind of leaving things open. If something's really controversial, we probably wait with the annotation and don't annotate it yet. We could get in contact with the authors if it was a request from someone. So there are lots of options for that. Another question, does complex portal also has the information regarding? Yes and no. Okay, information between SARS-CoV-2 and host. Right, you can see on here, let me just go back into the live page. So what we've done here, so you can see we're giving you access to the SARS-CoV-2 and SARS complexes directly. So if you browse here, you find the interaction with ACE2 and a link to Intact. And in Intact, we've done the same. We've given you a link to all the SARS-CoV interactions, and the coronavirus data set is probably what you're after. This is the data set of all publications the team, the wider team, the IMEX curator team, have annotated of coronavirus interactions within the virus, between virus and host, and associated host interactions as they're part of the publication. This is publication-centric, so this is IMEX curation. I don't know if that answers the question. Please write if you need a follow-up. Great. I think that's pretty much the end of my slides. Here's a link with a list of helpful links. The slides will be shared afterwards, so you can have a look at the links in your own time. This webinar, as well as a quick tour, are always available on the training website, and you have to filter by systems in order to get to the complex portal webinars. And I'll finish my slides off with a big thank you to the team, to our team leader Henning, the team coordinator Pablo, my fellow curators Livia, Sandra, Hema, and Edith, our developers Noe, Anjali, and Ellie, and our funders, which is EMBL-EBI, Open Targets, Wellcome Trust, BBSSC, and the NIH. Okay, so there was one more question about the protein structures that have now been identified by AI. We focus on anything that has experimental evidence. So we're not bringing in anything predicted. There are, I mean, for many years, people have predicted both interactions and complexes. And our focus, both in Intact and the complex portal, has always been on the experimental side. And we're leaving other databases to deal with the computational predictions. Right, I will switch my video on and see if anyone else has got any more questions. But thank you for great questions. That was really good. And I hope you enjoyed the jumping around and lively presentation. Thank you for your attention and interactions.